For 3 decades, the pharmacology of Alzheimer's disease has treated the lipid ceramide as a monotonic poison. The literature describes a consistent, alarming profile. As the disease progresses, ceramide accumulates in vulnerable brain tissue. It drives oxidative injury, permeabilizes mitochondria to induce apoptosis, and actively packages the vesicles that carry pathological tau from one neuron to the next. The therapeutic inference drawn from this data is straightforward. To protect the brain, ceramide must be pharmacologically lowered. That approach relies on a critical assumption, that ceramide has a safe, off position. It treats the molecule like an intruding pathogen that the healthy brain simply does without. The brain is actually the most sphingolipid-dependent organ in the human body. As we can see here, the myelin sheath wrapping this neuronal axon is a massive structural investment, built almost entirely out of complex lipids. Ceramide is the required foundational building block for that myelin structure, and its derivatives are heavily involved in executing synaptic transmission. Stripping a load-bearing structural lipid out of the nervous system indiscriminately risks accelerating the exact neurodegeneration it is intended to cure. Ceramide is not a biological dead end. It is an obligatory metabolic crossroads. This diagram models the cellular sphingolipid economy. The slow de novo road is the rate-limiting entry point. The sphingomyelinase road is a rapid response signaling route. Once in the hub, the cell directs ceramide outward into 4 routes to build structural components or survival signals. To build any complex brain lipid, the cell has to process material forward through this exact intersection. To degrade or recycle those same lipids, the cell has to send them backward through the exact same hub. Manipulating this pathway alters the entire lipid economy of the cell. A cell stripped of ceramide is a cell devoid of complex sphingolipids. In the central nervous system, that is an unsurvivable state. We can test the hypothesis that ceramide is purely toxic by looking at human genetic data. If lowering ceramide protects the brain, then genetic mutations that impair its synthesis should result in highly resilient, neuroprotected individuals. This table charts inherited disorders. When patients carry a SIRS1 mutation, synthesizing specific ceramide species is impaired, directly causing progressive myoclonic epilepsy. Altering a desaturation step in the DEGS1 gene causes a precursor lipid to accumulate, resulting in hypomyelinating leukodystrophy. These genetic lesions do not create a safe baseline. They produce fatal neurological diseases. Mutations in ASAH1, which cause ceramide to accumulate, are also neurodegenerative. Both extremes are severely pathogenic. The ceramide pathway possesses a strict biological floor. There is no monotonic axis where driving the lipid lower automatically guarantees safety. Empirical tissue measurements from human subjects at the earliest clinically recognizable stages of Alzheimer's disease confirm this structural dependence. In 2002, Hahn and colleagues mapped the single largest lipid change in the early Alzheimer's brain. This chart visualizes the findings. At very mild dementia, sulfatide, a critical myelin lipid, drops by up to 93% in gray matter, while other lipids remain flat. Simultaneously, sphingosine-1-phosphate plummets. The brain undergoes a dual loss of structural integrity and survival signaling. Transcriptomic data corroborates this physical loss. The genes responsible for glycosphingolipid synthesis are actively downregulated as early as mild dementia. The famous ceramide accumulation measured in the white matter of Alzheimer's patients is largely a passive byproduct. Traffic backs up at the metabolic intersection because the structural exits are closed. Artificially inducing this exact sulfatide deficiency in adult mice is sufficient to trigger chronic neuroinflammation and microglial activation without any prior amyloid pathology. The early Alzheimer's brain isn't suffering from a toxic overproduction of ceramide. It is suffering from a targeted structural starvation that forces ceramide to pool. Standard lipidomics measurements often rely on bulk tissue homogenates. Blending millions of cells together produces a single total ceramide number, resulting in highly contradictory literature. To extract scientific meaning, ceramide measurements must be mapped across 4 coordinates. First, species. C18 ceramide is specific to neurons, while very long-chain ceramides are specific to myelin. Second, compartment. Ceramide restructures whatever local membrane it touches. Generating ceramide at the plasma membrane to successfully bud an exosome is biologically distinct from generating it at the mitochondria to permeabilize the organelle. Third, cell type. Consider the conflicting pharmacological data regarding NSM-ACE2 inhibitors, which block a specific ceramide-producing enzyme. When an inhibitor was engineered to selectively target microglia, it successfully halted the propagation of pathological tau in mouse models. But when the exact same enzyme inhibitor was applied to general circulation in healthy mice, it disrupted the secretory pathway and severely impaired spatial memory. Fourth, time. Sphingolipids accumulate in the preclinical phase of the disease, but this gives way to frank lipolysis and membrane breakdown at the dementia stage. A single bulk ceramide number collapses these opposing cellular realities. It creates noise that obscures the actual mechanisms of harm. We return to the field's dominant therapeutic inference, that ceramide must be lowered in Alzheimer's disease. That instinct is directionally correct, but applying it bluntly is highly dangerous. The true Alzheimer's lipid pathology is a specific misallocation of metabolic resources. The cell directs less flux towards survival signaling, less flux towards structural maintenance, and allows localized toxic pooling. Genetic risk factors, particularly APOE4, tightly couple to this dysregulation, modifying these lipid states dynamically as the disease progresses. Future pharmacological interventions must respect the biological floor established by human genetic deficiency data. Successful therapies will need to target specific enzyme isoforms, isolated to specific cellular compartments within highly specific cell types. Treating Alzheimer's disease through the sphingolipid pathway is not a matter of shutting down the traffic, it is a matter of rerouting the intersection.